Okay. So I already gave you the definitions. Who is the father of genetics?
Gregor Mendel.
Gregor
Mendel.
Mendel. And then there's another word that's called locus. That's the location of a gene on a chromosome. This is a locus, this is a locus, that's another locus, locus. Locus for location. Try to remember that. I know I gave you guys a few quizzes. I want to know if you guys understand what true breeding or pure breeding means. Maybe tell me what genotype would you have? You can use any example for a true breeding plant. Because this might come up again if you don't understand what true breeding or pure breeding means, you will set up your Punnett square wrong. I don't know who's talking, but there's it's breaking up. I can't hear you. So Kawa said in the chat, maybe he's using letter P. Big P would be for one pure breeding plant, and little p for another characteristic of the pure breeding plant. So what this means, if it's a pure breeding or true breeding, that means it's homozygous, either for the capital letters, capital genotypes, or the lowercase. Anytime you see true breeding or pure breeding, you want to write the letters that are homozygous. Homozygous means the same, either uppercase, or lowercase, like how I wrote in the chat. And then you can go ahead and set up your Punnett square. If you had to define dominant versus recessive trait, how would you do that?
Dominant trait is always presented and the recessive is not.
So dominant trait takes over, not trait, actually I would say genotype. Trait is more of a phenotype. The dominant allele is going to take over, it's going to mask the effect of recessive allele, even though you might have a recessive allele for attached earlobes, but if you have the dominant one, that's the one that gets expressed. And then what does it mean to be homozygous dominant, homozygous recessive, and then heterozygous? That's still Mendelian genetics.
Homozygous dominant means it has like two of the dominant genes. Homozygous recessive means it has two of the recessive genes. And then what was the other one?
Heterozygous.
Heterozygous means it has one dominant and one recessive.
So like Daniel said, homozygous dominant, two dominant genes, homozygous recessive, two recessive genes, and heterozygous has two different ones, dominant and recessive. Another thing I want to mention for pure breeding, it's always the parental generation. So it's P crossed with P, that's the pure breeding. After you cross them, you're gonna get a hybrid. That's your F1 generation. I'm hoping, I'm not gonna do a cross Punnett square. I'm not going to give you any dihybrid cross. I'm only going to give you monohybrid, but I want to know, hopefully everybody can do a monohybrid cross. You had practice in the lab, you took quizzes, everybody can do a monohybrid cross. Can you guys set it up? Yes? Devin, Nancy, Daniel, Frene, Kaylin, Truce, Oscar. Yes, okay, so I'm seeing yes from everybody. So I'm not gonna go over that. Okay, and then we went over all the things that break Mendelian genetics laws. So one thing we mentioned was the linked genes. So this is showing independent assortment or law of segregation, actually, both. So this is showing that genes are packaged into different gametes, and this gamete is not affected by this gamete or this one or vice versa. So they are not affected by each other. They're separated into gametes. That's the law of segregation. So just remember the law of segregation. Now, the things that break this law, one thing we talked about was linked genes. Can you guys remember what linked genes were? What are linked genes? I gave you example of your appendix and your eyeball.
It's where one chromosome, not chromosome, I'm sorry, one uh, sequence of DNA has an effect on another one, like even though they're different.
So their linked genes means different genes are located on the same chromosome, and if they're really close to each other, there is no crossing over happening. If you have one gene, you're gonna have the other gene because they are on the same chromosome. If we go back here for the independent assortment, that means one gene is located on one chromosome and they can separate. They can separate here. But if it's two genes on the same chromosomes, that means they're linked and they cannot separate. And that doesn't follow Mendelian genetics or the law of independent assortment. Also, one more thing. This one is dihybrid actually here. So Punnett squares are used to track different genetic disorders. I will give you some questions about this, especially with the X-linked that we talked about. Do you guys understand what a carrier means? So if the problem says this parent is a carrier for this, do you understand what that means?
It means they have the mutation or they carry it. Like I don't know how else to say it.
So they have one gene that's affected and they carry that gene, but they also have a healthy gene. So they don't express the affected gene. So they have one affected gene and they have the other healthy gene. That means they're carriers for it. Then we have incomplete dominance, codominance, and pleiotropy. Someone help me out with that. What are those?
Incomplete is when you have kind of 50-50 reflection.
You have a
Sort
mixture.
of, yeah, mixture, yeah, such as mixing blue and red to give
Okay.
you purple.
What about codominance? Who wants to take on that one? Oscar.
Co is
Yeah.
a combined effort.
Combined. Kaylana, were you about to say something?
I was just going to say that both of the phenotypes are presented equally.
Genotypes. So both of the dominant genes are expressed equally, and I gave you example for your blood type. What is pleiotropy? Pranay, you're talking, but I can't hear you.
Is it one gene has multiple functions?
One gene has multiple effects
Okay.
or multiple biochemical pathways. So one gene can control different pathways or different effects. For the incomplete dominance, I gave you this example for snapdragons, and blood type is codominance. And then for this one, I gave you example of sickle cell anemia. So if I say, oh, this person has sickle cell anemia, is what type of, you know, which one would you apply? Pleiotrophy, incomplete dominance, codominance. So try to remember the examples I gave you. And then you can expect some X-linked recessive disorder or X-linked disorder questions. And Kawa in the chat said, when someone is a carrier, they are hetero. That is true. They're healthy and affected both. And then another thing is if I say, maybe tell me the percentage of the sons that are healthy, you only look at the sons. If I say daughters, you only look at the daughters. If I say all offspring, then you look at the whole box. So try to pay attention, try to read the question multiple times. You don't have to memorize any of these, the charts, but these are the most common ones. Last thing you guys have is the X activation. I gave you example of CC the cat. It's a carbon copy of a cat. They took some skin cells from a cat, they grew another cat. But this original cat was all, I think, black fur, and this one has some orange fur. That's because it has alleles for black fur and the orange fur. but because of the environment, black fur got activated here and orange got activated here. So it has different colors of the fur compared to the original, even though it has same genetic information, which means it has genes for both. The reason it might be environmental, maybe this cat lives in a really cold place and black color makes it a little bit, easier to be in the cold and gets activated. Same thing for the Siamese cat. It says environment can alter phenotypes. It's black in the paws and the tail and the face because it's cold in those areas. But if this cat was living maybe in an area where it snows a lot, it's really, really cold, maybe it'd be all black fur on this cat. And then the last thing you guys have is polygenic. What is polygenic? Can you guys give me an example?
Polygenic is when a gene is being controlled, right?
Polygenic is no, somebody else wants to go. Poly means multiple. Genic means gene.
When the phenotype is affected by more than one gene.
One phenotype is affected by more than one gene. The example I gave you was skin color. Another one would be eye color. You have lighter skin color, you have dark skin color, you have everything in between. Same thing for the eyes, you have multiple colors because there's multiple genes controlling that. That's chapter 10.